Welcome dear viewers to the video on stability failure of pharmaceuticals and significant change. The video will be very important as this video will cover the stability failure reasons, how the stability sequences are designed and what is significant change. So let's start with the video. First of all, we will see the basics. When you can say that the product is stable or when you can say that product is not stable. So I have considered USP chapter for this definition and information. The product which meets the predetermined specification throughout the expiry period or shelf life period is a stable product. The simple meaning of this definition is the product which meets CQS as at initial and during the shelf life period. That means the product retains the quality and purity throughout the stability period, and the product is safe to be consumed throughout this period. For example, you have prepared a batch of tablet, for example, you have tested it and you loaded it on stability. You have some specifications for assay dissolution, water content, disintegration and the impurities. So you have tested and loaded it on the stability. Now after 6 months, after 1 year, after 2 years, it meets the predetermined specifications which were at the initial level. So that time you can say that the product is stable. That means in simple words, the product is same as on the manufacturing date and the product is same in between any time of the expiry period. You have given, for example, an expiry period of 2 years for the formulation. So the formulation will pass the criteria or pass the all CQS like initial testing. That means the potency will be maintained, the dissolution profile will be maintained, the physical appearance and the impurity limits will be passed by that formulation, or the impurity will be in the predetermined specification. So this is the simple meaning of stable product. And if any of these criteria is failing, that time you can say that the product is failing or product is not stable. Now what is the definition of stability as per USP? Stability is the extent to which a product retains within the specified limits and throughout its period of storage and use the same properties and characteristics that it possessed at the time of its manufacture. That means as it was passing at initial stage, still that product pass at any point of time in the expiry period. Also, one thing here is to consider and to remember is that this stability is not the in-use stability. This definition is not for the in-use stability, it is for the expiry period or shelf life period stability. In-use stability is different. Many of the stability parameters, that means critical quality attributes, CQS, are also dosage form dependent apart from the basic CQS. Like you have prepared a tablet with DT of around 4 minutes at the initial time point. So will it be maintaining that DT for 4 minutes or not? Or that DT will change? So certainly that DT may change by a little time like 1 minute or 2 minutes. But still it will pass as the dissolution is meeting. So that is very minor change and it will not be considered as a stability failure. Now what are the types of stability? So stability is physical stability, chemical stability, microbiological stability, therapeutic stability and toxicological stability. If the product meets all these stabilities, then the product is said to be stable, and the patient can consume that product within the shelf life period. The product after consumption will be safe and will be having the efficacy. Chemical stability means each active ingredient retains its chemical integrity and level potency within the specified limits. Simply you can remember it as assay or potency. Physical stability means the original physical properties including appearance, palatability, uniformity, resolution and suspendability are retained. Microbiological means sterility or resistance to microbial growth is retained according to the specified requirements. Antimicrobial agents that are present retain effectiveness within the specified limits. Therapeutic stability, the therapeutic effect remains unchanged. Toxicological stability, no significant increase in the toxicity occurs. So this information I have taken from USP general chapter, and this is very basic part, and it is very much required for the understanding of stability. Stability quality control or stability critical quality attributes for tablets and capsules I have considered here for example. Consider a formulation of tablet or capsule. So you will require to have specification for description, appearance, assay that is potency or label claim, dissolution, organic impurities or related substances, and the microbial limits. Along with these basic CQAs, other CQAs like disintegration time, hardness, any impurity if you want to monitor it during stability, and loss on drying and water content. So based on the formulation, these other tests may vary. Now what is significant change? So many of the professionals don't understand about the significant change, and also many of the professionals found this topic as confusing. So simply significant change means non-conformance to the quality parameters or not meeting the predetermined specifications in the stability period. So significant change will affect the safety and efficacy or palatability. So whatever the changes or failures are there, if these failures are affecting the safety and efficacy, then you can say that the change is significant. Like example, as per ICH guidelines, these examples are given for the significant change. Change in the assay or potency by 5% from the initial, or failure to meet the assay and potency criteria. Sometimes there is a change in the assay due to analytical method variation or due to analytical error. That time the sample may be tested second time, and if it is passing, then it will go into the OOS or OOT category, and it will not be a significant change. So OS and OOT category is a different topic. Here we are considering only the significant change. But if after repeated testing also you found that there is a decrease in assay, and if it is reflected by the mass balance, that is increase in the degradation, then it will be a significant change. Failure to meet dissolution test or dissolution specification on 12 units. So that is S2 stage. First is S1 stage and second will be S2 stage for IR formulations. Failure to meet the limit of impurity. If impurity increases, that time it will be a significant change. But still, if impurity specification is given for example as a 0.2% and you found impurity at accelerated condition like 0.3%, that time it will not be a significant change. You will go to test the intermediate condition samples. Also sometimes the impurity qualification go in parallel with the stability studies, and if you found that the impurity qualification limit is increased due to qualification of the impurity, then it should not be considered as a significant change. So that is also a different topic, but always understand what can be the significant change and what will not be the significant change. Then change in the physical attributes will be a significant change. Failure to meet the pH limits for some liquid formulations, that may be oral liquid or sterile liquids. And in simple meaning, failure to meet the CQS, critical quality attributes, will be a significant change. Now we will see what will not be a significant change, or what may not be considered, or which failures may not be considered as a significant change. So if the changes are only due to the nature of the formulation, or due to the storage of the formulation at accelerated condition, considering the formulation design and the API properties, that time the change will not be considered as significant. The example is change in the physical attributes of suppositories, like melting of the suppositories, melting or phase separation of the creams, phase separation of emulsions. Sometimes the capsules may become may change color due to higher temperature. Sometimes the degradation may be due to the melting of the API at higher temperature for the low melting point APIs. That time the change will not be considered as a significant change. Also, dissolution failure due to gelatin cross-linking will not be considered as a significant change if the product passes the dissolution test with dissolution media containing the enzymes. If dissolution is performed on 6 units in the media without enzyme, and if the dissolution fails, then it will not be a failure. So this criteria will be clearly written down into the protocols, and as per the protocol remarks and precautions, the significant change will not occur or be considered. If significant change occurs at accelerated condition, then the testing to be done at intermediate storage conditions. This is very important to understand here, that accelerated condition is for 6 months, intermediate condition is for 12 months study, and long term is for 24 months or 36 months, or as per the applicant's requirement for the shelf life. So generally the change at significant change at accelerated condition is not considered. If that change occurs at accelerated condition, intermediate samples are taken out and those are analyzed. Also, if the product is not stable, or if the product is showing significant change at intermediate condition, then you can go for the long term condition and you see whether there is any failure in the CQS at long term condition. If product fails at long term condition, for example at 2 years, then there are different ways to deal with that formulation, which may be reformulation of the formulation, or shortening of the expiry period, or there may be a chance that the product is required to be stored at refrigerated condition of 2 to 8 degrees Celsius. So that is different part. It is not a part of significant change, but for your understanding I am explaining these things. Now we will see the failure in the stabilities. When you can say that the product is failing, and I have considered the CQAs like description, assay, dissolution, impurities, and the microbial attributes. Also, I will include the best practices to avoid the stability failures. So, description. Description is the product appearance, and change in the description due to any physical or chemical changes will be a failure. Spots on the tablet surface. Tablet lamination on storage. Tablet breaks on storage, that will be a failure. Physical and chemical incompatibility of the drug and excipients. This might be the reason, like Maillard reaction with the lactose. Change in the capsule shell color. softening of capsule shell of hard gelatin capsules, leak in the soft gelatin capsules, change in the color of oral liquids and change in the pH, layer separation in multilayer tablets, hard cake formation in suspension or change in the API pHD in the suspension formulation, recrystallization in the suspension formulation or recrystallization or formation of insoluble crystals in solution dosage form. So these changes may occur, the reasons may be different, sometimes the reason may be improper storage. Then assay or potency failure, increase or decrease in the assay or potency. So increase in the assay may occur due to loss in the liquid vehicle or water in semi-permeable container formulation. If the formulation is stored in semi-permeable container formulation, that time the product may get uh, analyzed and with results may come as higher assay. So that is because loss of vehicle. Sometimes decrease in the assay. Low or high content of API in the formulation may be the reason for analytical variation. Increase in assay in liquid formulation due to loss in the vehicle or water. Increase in assay due to drug degradation. Sorry, this is decrease in the assay due to drug degradation. And this decrease in assay due to degradation should be mass balance with the impurity. Loss in API. Some APIs may be volatile. For example, I have considered here as a reason. Absorption of API in the container or closures. Microbial reasons. Assay and potency calculation and label claims. Sometimes there may be a mistake or a error in assay or potency calculation, or sometimes there may be a mistake in label claim. So these may be the problems for assay and potency failure. Problems in the blend uniformity and content uniformity may lead to failure in the assay or may lead to analytical variations. Use of low level of stabilizer or not using the stabilizer at all. This may be one of the reasons for the formulations or APIs which require some sort of stabilizers. If you don't use the stabilizers, then API will get degraded. So that is due to the improper design of the formulation. Then coming to the another CQA, that is disintegration time or dissolution failure. So generally disintegration time is not fixed uh, before the stability studies. Dissolution criteria is required to be specified, but here I have considered both. So this change in disintegration or dissolution may happen due to hardening or softening of the formulation, which may be tablet or capsule. Change in the API form. If you are using crystalline API and if the form gets changed and that form is less soluble or insoluble, it will lead to decrease in the dissolution. Moisture absorption or moisture loss, change in the formulation hardness, loss of disintegrant activity, gelatin cross-linking, improper specifications of dissolution sometimes may lead to dissolution failure. So always whenever you are going for stability studies, have a dissolution profile testing. Include all the time points and test the formulation so that you will avoid the significant change, and as per the data you can set the specification limits after completion of the stability period. Sometimes pack integrity problem may lead to dissolution and disintegration problems. Then related substances or organic impurity failure. Failure or increase in the impurity in the formulation. That may be due to the drug degradation, that may be due to other reasons. So drug degradation or stability due to the manufacturing process may also happen. Degradation due to the incompatibility of excipients or other APIs in the combination formulation. Sometimes the combination formulations are there in which one API is low in content while the other may be high in content, and due to the incompatibility between these two APIs, one API degradation may happen. The incompatibility may be due to the excipients. The process impurity in the API may be a degradant in the formulation. Sometimes if you consider a process impurity and you consider a process impurity in the API and you don't consider in the finished product, And if that process impurity is also degradant impurity at that time, you will get surprise with that process impurity is getting increased during the stability. So fixing the impurities and impurity specification is very critical, and that may lead to increase in the organic impurity if specifications are not proper. Sometimes formation of adducts or dimers of the API may happen, and that may lead to increase in the impurity. Then formation of adduct of the API and excipient may also be possible for some of the APIs. Interaction with the container closure system may happen, which leads to increase in the impurity. This happens generally with the formulations which are liquid in nature. Then other reasons like loss in the stabilizer content or effectiveness of the stabilizer, change in the API form, change from the initial form to the less soluble form, or change to the form which is having more degradation or which is prone to degradation. Chemical incompatibility of the drugs and excipients may also lead to increase in the impurity limits. Impurities in the raw materials and the APIs. Sometimes this becomes the very critical for nitrosamine type of impurities. If your APIs are contaminated with nitrite or nitrites, if your excipients are contaminated with the nitrosetting agents, that time these impurities may create a big problem. Water content and water activity of the raw materials. This is very critical for the APIs which are prone to hydrolysis or sensitive to the water content. In process materials, finished product and the stored formulation. Sometimes the formulation may absorb water from the environment, and if packaging system is not capable to protect it from the moisture ingress, that time the impurity formation may happen due to the hydrolysis of API due to water increase. Change in the pH of liquid formulation may also happen, and which leads to the impurity formation. Acidic and basic components of the formulation may also lead to the impurity formation if the API is sensitive to some specific pH range. Then microbial contamination or microbial failure. Generally, microbial failure is a very critical thing for sterile formulations. Also, some of the oral solid formulations and topical formulations may get contaminated, and that contamination may be due to the problems in the container closure or contamination that may lead to the increase in the microbial load. And sometimes formulation composition might be such that it uh, is prone to the microbial load. Then pack integrity may have some impact onto the microbial failure. Storage conditions and time of exposure. So sometimes if the product is exposed to the temperature which is higher than the storage temperature, that means temperature excursions beyond certain limits or beyond certain time, then that may be a problem in increasing the impurity or significant failure. Humidity excursions may play a significant role. Light exposure may lead to photodegradation. Oxygen exposure lead to oxidation of the API and formation of impurities. Humidity exposure lead to moisture ingress and hydrolysis of the APIs. Packaging defects and packaging process defects may also lead to the significant failures. Like you are performing packaging of blisters and the blister, some of the blisters fail in leak test. So that formulation will be very much prone to the moisture ingress, or if sealing is not properly in bottle formulation. So these things may lead to the significant failures. Best practices. Now, if you are going to develop a formulation and you don't want that you will get the surprises for stability failures, so some best practices are enlisted here. Studies to understand the intrinsic stability of the API. Whenever you get the API in your hand, you try to understand what is the intrinsic sensitivity of the molecule and intrinsic stability of the molecule. Selection of suitable manufacturing process. If you find that the molecule is sensitive to water or is sensitive to drying temperature, then go for dry granulation. Protect it from light and humidity if it is sensitive. Evaluate the whole time so that the impurity formation will be restricted. Storage conditions. Establish the particular storage conditions and packs to store the API and the excipients along with the raw materials of in-process stage. Study the polymorphism API in API and check whether the polymorph change can occur. Then understand the potential degradation mechanism and the impurity formation pathways. Perform force degradation of API along with the formulation. Perform extensive compatibility studies. Stress testing of the formulation by storage at harsher conditions. For example, if you want to do the study, store the formulation for some weeks or some months at 50 degrees to understand whether there is an impact of temperature. Then stability of the batches with different compositions. Load the stability batches made with different raw materials or compositions to understand the problematic excipient or problematic process. Stability in different packaging systems. Always have a stability data with different packaging systems like aluminum blister, PVC blister, PVC PVDC blister, acryl blister, and many more. In HDPE bottle, have a data with low weight HDPE bottle, heavy weight HDPE bottle, and HDPE bottles with desiccants. Suitability of the packaging materials to be evaluated mainly for the liquid formulations. Then you should always have a stability indicating analytical method whenever you are going to perform these studies. If the method is getting changed or updated or revised, then recheck the formulations which are prototype and which formulations you are going to finalize. Always have the stability data of final formulation composition in the final packaging system. So these are some of the best practices. And stability, as we consider, we know the regulatory requirements, we know the protocols, we know these things, and we uh, have good understanding about those things, but we fail to understand the reason behind the stability failures. So my motto of preparing this video was to have good understanding about the reasons behind stability failures. I hope you will understand these things and you will apply those things in your uh, professional work. So thanks for watching the video. Please do like, share and subscribe to Pharma Learning in Depth channel. Thank you.